Hello my dear students, welcome to our channel Rajesh Biology and I am Rajesh, MSc Zoology. Today our topic is gastrulation. That means before that also we are going to discuss about the bilaminar embryonic disc, how the bilaminar embryonic disc convert into the trilaminar embryonic disc by the process of gastrulation. This is I am going to discuss in this chapter, in this class. Okay, not only the gastrulation, neurulation also. So first we will start from the implantation process. Here if you observe, the implantation process we already completed in the previous class. Okay, that implantation starts after the 6th day of fertilization. Once the fertilization is completed in the January 1st, for example, fertilization is completed. January 6th or January 7th, the implantation process will be begin. Okay, and by sending that syncytial trophoblast into the endometrium and by getting the nutrients, and it develops trophoblastic villi. Okay, cytotrophoblast, syncytial trophoblast, completely embedded within the uterine wall. It takes 1 week time. That means at the end of the second week, the implantation process will be completed. The process of implantation will be completed at the second week after the fertilization. That means nearly 13th day, 14th, January 13th, January 14th, if fertilization takes place at the 1st January. Okay, come back. So in previous video, just did a small mistake. That is what the inner cell mass is towards the uterine wall. Where there is an inner cell mass, on that side only implantation takes place. On other side there is a blastocele. Okay, this is implantation. Now in this video I am going to explain what will happen to the blastocyst after implantation. What will happen to that embryo? This is called embryo up to the 8th week of development. Still it is 2nd week only, right? Okay, up to the 8th week we call it as a embryo. From the 9th week only we call it as a fetus. So in this embryonic development what will happen? This is outermost, this green color lining is syncytial trophoblast and the inner what? Cytotrophoblast. Okay, and this blue color, this is embryonic proper, which has the stem cells. Stem cells means which can develop into pluripotent stem cells, which can develop into the endoderm, ectoderm, mesoderm also. Okay, all the stem cells undifferentiated, still it is not differentiated. Okay, undifferentiated stem cells are present here. This is embryonic proper. And this cavity is called blastocele, in which that it is also considered as the primary body cavity. Okay, this is the situation, and it is completely implanted. It is completely embedded. It is embedded within the uterine wall. Then I am taking this only. That means only the trophoblast and inside the inner cell mass. From now onwards I am explaining this. Okay, bilaminar embryonic disc, how the bilaminar embryonic disc is formed. Okay, the cells first differentiated into the two layers. This is the first differentiation. Okay, up to now all cells are equal, but later they are adjusted. They are adjusted in two layers like this. One is blue color layer, another one is purple color layer. I draw here. Okay, so like these two layers they adjusted. One layer which is outer side, that layer is called epiblast. That is called what? Epiblast. And another layer of cells which faces the cavity, which faces the blastocele, that is called hypoblast. Epiblast is the layer of cells which faces the villi, which faces the syncytial trophoblast, that is called epiblast, outside. Inside, which covers, which lines the blastocele, which faces the blastocele, that is called what? Hypoblast. Okay, that is the first thing. Like this, first differentiation of cells takes place. After this, what happened? This hypoblast lines the cavity and it get delamination. Delamination means what? You know lamination process? Lamination process, for example, if you take your Aadhaar card to any Xerox center or some lamination centers, they will keep two covers. In between two covers, they keep one Aadhaar card, and in that lamination method, both are attached with one another. They both come closer. Okay, this is called lamination. Delamination means already attached layers get separated. This separation is called delamination. How this delamination process takes place? For example, if it is the trophoblast. In this trophoblast, the cells embryonic proper is actually attached like this. Okay. After delamination process, what will happen to this? In the delamination process, they will move away from the trophoblast layer. They come into the middle like this. What happened? Already attached cells get delaminated. They are separated. They comes down. Okay, like this, they are delaminated, and the blastocele cavity is spread throughout this embryo. Within this, it will spread like this. Okay. Okay, within this also delamination process takes place like that. One cavity is formed. Okay, now this stage is called bilaminar embryonic disc. Why it is called bilaminar? Because here there is two layers. Before differentiation, there is only one embryonic cell proper, okay, or inner cell mass. There is no cell differentiation. Once the cells get differentiated into the epiblast and hypoblast, two layers of cells is there. That's why it is called bilaminar. Still it is before the 9th week, so it is the embryo, bilaminar embryonic, and it is in the disc shape. You know, DVD, CD, how they look like. As like that, the cells are arranged in this manner. Okay, and it is covered by the trophoblast. Okay, it is attached to the trophoblast. You can see like this. For example, if it is the trophoblast, this red color lining, what it is? Trophoblast. Okay, with the trophoblast also there is some cells is there, and these cells are attached like this in this two-layer manner. So bilaminar embryonic disc shape. So to show that disc, just try to draw a 3D diagram in this manner. You can observe this middle part. This middle is the disc-like structure, and that is the covering. So it is called bilaminar embryonic disc. Still the gastrulation process is not yet started. Okay, before it starts, you have to know what is gastrulation means. Now, gastrulation is a dynamic process. Gastrulation is what? Dynamic process. Dynamics means movement, which is in the locomotion, which is in the movement. That is only we call as a dynamics. Okay, so gastrulation is a dynamic process, means cells are moving. Okay, so when the cells are moving, the gastrulation process will be start. Because of the movement of the cells, because of the gastrulation of the cells, bilaminar embryonic disc convert into the trilaminar embryonic disc. So gastrulation is a simple migration of cells, movement of cells is called gastrulation. How the gastrulation takes place? In this embryonic disc, I am taking this embryonic disc out, I am keeping here. This is the embryonic disc. Okay, so in this embryonic disc, in a longitudinal axis, one streak is formed. One streak. Okay, the primitive streak, it is called what? Primitive streak.